Good morning, students. Welcome to Kendriya Vidyalaya Sangathan online classes. We will be discussing about sexual reproduction in flowering plant in this chapter. This chapter is divided into 7 different sessions from session 1 to session 7. This is session number 1 and we will be talking about the flower, the various pre-fertilization structure and event, the stamen, microsporangium and pollen grain, structure of microsporangium, microsporogenesis, the pollen grain and viability of pollen grain. Rest of the chapters will be taken up in the next few sessions. After doing the chapter, you will be able to define a flower, draw and label various parts of the flower, differentiate between male and female reproductive structure, get an exposure to the different pre-fertilization events and structure, draw and label a stamen, microsporangium and pollen grain, draw and label the structure of microsporangium, appreciate the process of microsporogenesis, appreciate the pollen grain, and also to know about the viability of the pollen grains. This is quite an easy chapter for you. Why? Because class 11th you have done some of the important things like anatomy of flower, various terms associated with description of typical flower, term associated with different modification of androecium as well as gynoecium. So what is a flower? We know that flower is the reproductive part of a plant. It is defined as the part of the plant that is often brightly colored and provides the pollen that is moved from plant to plant for pollination. The botanical definition is the flower is the seed bearing part of the plant consisting of reproductive organs, stamen and carpel. They are typically surrounded by a brightly colored corolla and green calyx. You can see the bonanza of different type of flowers which are present all around us, clicked here and there through my camera. You love it. Why flower? Because they are object of aesthetic, ornamental, social, religious and cultural value. We love flowers. This is the diagram of a typical flower and you can see that there is an gynoecium in the center followed by androecium, then comes the petals, call them corolla, and the sepals, call them the calyx. At the base there is thalamus. So a flower, typical flower has got 4 different roles as you already know. What are the different pre-fertilization structure and event? We know that in the flower the androecium and gynoecium are the reproductive structure. They differentiate and develop. The androecium consists of rolls of stamen representing the male reproductive organ and the gynoecium represent the female reproductive organs. How does a flower develop? A flower develops from a floral primordium by different hormonal and structural changes. What is the inflorescence then? It is the arrangement of flower on the axis. Let us have some discussion on the stamen, the microsporangium and the pollen grain. A typical stamen has got 2 different parts, the filament as well as the anther. They are connected to each other by different ways in different plant species. It can be fixed at the base, it can be fixed at the back, it can be extended the entire length, and typical terms can be dorsifast, basifast, adenate, versatile. Class 11th, if you have studied a little bit beyond the book, you might have come across those terms. The number of the anther varies from flower to flower. A typical anther is bilobed and dithecous. You need to know about these 2 terms. Bilobed means 2 lobes and dithecous means there are more than there are 4 lobes. Theca. Theca means cover. There is a longitudinal growth that runs the entire length, and it has got 4-sided tetragonal structure consisting of 4 microsporangium which are located at the 4 corners. If you can see the diagram, these are the 4 pollen sacs. The microsporangia develop to become pollen sac that extend longitudinally through the entire length and they are packed with pollen grains later on. The microsporangia develop further and become pollen sac. This is something for you to remember. When we talk about the theca, there are 4 important theca. Theca are the cover, 4 walls. From outside in, it is the epidermis, then come endothecium, middle layer and tapetum. We need to know the function. The epidermis is basically meant for protection and they help in dehiscence of anther and release of pollen. whereas the tapetum, the innermost layer, they are important for providing nourishment to the developing grains, pollen grains, and the cells are laden with dense cytoplasm, and generally they have more than one nucleus. In the anther, the cells of the sporogenous tissue undergo meiotic division to form microspore tetrad. The set of four is called tetrad, which is the potential pollen or microscope mother cell. So the term PMC stands for pollen mother cell. So what is microsporogenesis then? Microsporogenesis is the process of formation of microspore from a pollen grain through meiosis. What is a tetrad? Microspore tetrad. The microspore tetrad in a cluster is a cluster of anther mature and dehydrate. The microspore disassociate from each other and develop into pollen grains. Naturally, the term dehiscence is a new terminology. Dehisce is to get released. Inside the microsporidium, several thousand of microspore or pollen grains are formed that are released with the dehiscence of anther. Let us have a look about the transverse section of mature anther. If you can see this diagram, you can see that there are four different layers. And the other side, you can see a magnified view of the four layers. Outside in, it is epidermis, followed by endothecium, then comes middle layer, and finally the tapetum, which is a multi-layered structure. You can see the cells are big, laden with a lot of cytoplasm, and it can have more than one nucleus. And you remember the function, the first three layers for protection, and the last layer is basically meant for providing nourishment. It is a mature anther. You can see the dehiscence slit at both the sides, and you can see the pollens are getting dehisced. Let us have some discussion about the pollen grains too. The pollen grains represent the male gametophyte. They are of beautiful shape, size, color, design when you observe them under microscope. Some mesmerizing gold, and the size is about 25 to 50 micrometer in diameter. The layer, it has got two important layers. The outer is called as exine, and the inner layer is called intine. The outer layer is made up of a very tough, resistant, rigid chemical called as sporopollenin. It is virtually the most important organic material that is known, resistant to high temperature and strong acids and alkali as well as enzyme. Means if a person or animal will eat the pollen, they cannot digest it because sporopollenin is not affected by enzyme. What a degree of resistance it has got. When it comes to the intine, it is made up of cellulose and pectin. It is thin and elastic. You might have come across the term germ pore earlier. The germ pore is a place where sporopollenin is absent. So when the pollen grain will germinate, the intine will come out in the form of a pollen tube carrying the male nucleus. You can see the diversity of different type of pollen grains. Now, when the pollen are formed, they are one cell stage, one nucleated stage. But during the process of maturation, the cell divides into two, and it has got two cells, the vegetative and the generative. The vegetative cell is bigger, has lot of food reserve and large irregularly shaped nucleus. The generative cell is small and floats in the cytoplasm of the vegetative cell. The nucleus is spindle shaped with dense cytoplasm, and when dehiscence will take place, in about 60% of the angiosperm, they are dehisced in this stage. But in the rest of the 40%, what happens? The generative cell again divides mitotically to give rise to two male gametes, that is the first male and the second male nucleus. So the first is the vegetative that transforms into the tube nucleus, followed by generative, which divides into the first and the second male nucleus. It becomes a 3-cell stage. So 40% of the angiosperm, they are released in 3-cell stage. This is the diagrammatic representation of how a pollen develops into a 2-cell stage. You can see this diagram number 1. It is one cell, one nucleated stage. Slowly what's happening, vacuolization is taking place, the cell is growing in size. Then this nucleus is expanding and a symmetric spindle formation will take place. And slowly over a period of time, the cell is divided into vegetative and generative cell. The generative cell floats almost inside the vegetative cell. You can see the nucleus. Vegetative cell nucleus is large and globular. Generative, it is spindle set. A matter of concern, are pollen all good? Answer is yes. Pollen can be highly beneficial, but at the same time they can cause a little bit of problem too. Pollen grains may cause chronic respiratory disorder like asthma, bronchitis, etc. Even skin allergy can be caused because of the pollen grains. For example, Parthenium or carrot grass that grows abundantly, it is an alien species invasion species that has come from other country, and because competitively it is superior, so it is growing here and there. So even the pollen that cause one of the major problems, that's asthma and bronchitis, in the places wherever it grows. Whenever it comes to the beneficial use of pollen grain, they are rich in nutrients. They can be used as food supplement, and the pollen product in the form of tablet and syrup are available in the market. And the most important, they are claimed to increase performance of athletics and racehorse. So it does have certain very interesting use. What about viability of pollen grain? First, let us clear up what is viability. From the time of dehiscence till they are able to fertilize, they are capable of fertilizing. That period is called as the viability period. And the viability period, obviously, they differ from species to species. That depends on the prevailing temperature and humidity, even on the quality of the pollen grain. For example, in some cereals such as rice and wheat, pollen grains lose viability within 30 minutes of their release. If they are able to pollinate, fine, otherwise they cannot anymore. In some species of Rosaceae, Leguminosae and Solanaceae, they maintain viability for months. So naturally the question is, can we increase the viability artificially? Answer is yes. Cryopreservation. Cryopreservation is a technique by which the pollens are stored in liquid nitrogen at minus 196 degrees centigrade, and later on they can be used for different crop breeding program. Let us recapitulate back. What are the different parts of flower? The androecium, gynoecium, the calyx and the corolla. Basically, the androecium and gynoecium because they are the reproductive part of the flower. What are the various pre-fertilization structure and event? We talk about the stamen. It has got two different parts, the anther and the filament. When we talk about the stamen, the microsporangium and pollen grain, a typical angiosperm anther is bilobed and dithecous, and the microsporangium develop and become pollen sac that extend longitudinally through their length and packed with pollen grains. What about the anther? Anther are four different layers. You have to recall epidermis, endothecium, middle layer and the tapetum. What about the microsporangium? In the mature anther, they are compactly placed as homogeneous tissue. They are also called as sporogenous tissue. And later on, by microsporogenesis, pollens are formed. The process of formation of microspore from a pollen mother cell by meiosis is called as microsporogenesis. They are in the set of four. Initially, that's how we got the term microspore tetrad. When you talk about the pollen grain, they represent the male gametophyte. There are a variety of architectural shapes, size, color, designs, and the size also varies from plant to plant from 25 to 15 micrometer in diameter. They have two prominent layers, the exine and the intine. What about a mature pollen? When in the two cell condition, they have got a vegetative cell and a generative cell. The vegetative cell is large, having globular nucleus. The generative, they have spindle subnucleus, dense cytoplasm. What about the pollen grain in human being? We know the pollen can be highly allergic. They can affect the respiratory system, causing asthma and bronchitis. Whereas they have rich use also. They can be used as nutrient. They can be used as pollen tablets, as food supplement. They are in the form of tablet and syrup. And even it increased the performance of athletics and racehorse. So it has got a lot of demand in the market. When it comes to viability of the pollen grain, The pollen grains viability period differs from species to species. Somewhere it is about 30 minutes, like in case of rice and wheat, whereas in some roses and all, that can be for months together. Artificially, the viability period can be increased by cryopreservation. Attempt the following question. Dash are formed when flower which bear the floral buds and then the flowers. Which will be the correct answer? I am leaving this for you. You have to attempt and then tell the correct answer. The male reproductive part of the plant is called what? Androsium, gynoecium, anther or filament. The female reproductive part of the flower is called androsium, gynoecium, anther or filament. Choose the correct term. The size of the pollen grain measures about how much micrometer in diameter? 25 to 50, 5 to 20, 100 to 200 or even more than 500 micron meters. Choose the correct one. Then you should also be able to tell me what is the full form of PMC. Also, what is the chemical composition of enzyme? So out of the 4, you have to find the correct one. MCQ. What is the chemical composition of enzyme? Again, out of these 4, choose the correct one. After you do it, you can compare your results. These are the results. These are the solutions. Find out how much you have scored. And now I want to give you some work, some home assignment. Try it back at home. You have to draw, label and describe a mature pollen grain, differential between exine and intine. Then you have to tell what are the various layers of anther, mention their function. Then what is the significance of pollen grain for human being, and then just the cross section of anther has been given. You have to label different parts. Some home assignments are also given to you. You can copy the link, put in the menu bar, or even you can click on them and attempt the different parts of typical flower. Some tests have been designed for you. You can try that. You can do the match the following. For further reading, a class book of botany by A.C. Datta is a good one. A lot of information is there. And obviously the NCERT textbook is one of the best things that can happen to you. You can read it from one side to other side to get a good grasp of the book about the concept. Some website links I have suggested for you. You can go through them, and some video links also. Please go through them and your concept about this topic will become much more beneficial. Thank you. So for the next session, we'll be discussing about pistil, megasporangium, embryo sac, the microsporogenesis and the female gametophyte. Meet you then. Thank you. I hope you enjoyed. Meet you for the second session. Keep watching.